There we go. Lesson 12, group assignment. Screen share. So problem 1 is just kind of some terms. Let's see if we can get that on there. Which of the terms describes or is an example of regulatory proteins? That would be activators, repressors, and CAP. The example of small effector molecules would be inducer, inhibitor, allolactase, and cyclic AMP. DNA sequences found are the +1, the -10, and the CAP. Which of the above is not involved in lac operon? I said all of them were involved in some way or another. They either activate each other or are activated because of it. They're a part of the process and each play an important role. The structural genes are LAC-Z and LAC-A.
Okay, number 2, more wonderful definitions and special terms page we all love. So in 2A, it's talking about the lac operon is regulated by 2 inducers. Those 2 reducers are repressors and activators. B is talking about binding sites for signal factor and RNA polymerase. So that binding site is called LAC, or the promoter, while the CAP site is the binding site for the activator that regulates the lac operon. Is in reference to the enzyme adenyl cyclase that converts ATP to cyclic AMP. The special sequences, the -35, -10, or the +1 sequences, are found within that LAC-P gene or portion of the lac operon, the promoter. The LAC-L gene encodes the LAC repressor, while the Y gene, the LAC-Y gene, encodes lactose permease. For F is talking about attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And G is talking about the E. coli strain that contains 2 copies of the lac operon, i.e. the murozygote strain. Yep.
Okay. So problem 3, let's see, is asking when the E. coli cells will be blue because of the X-gal molecule. So in the first case, IPTG is absent as well as glucose, but X-gal is present, but it will not occur because the reaction cannot occur. IPTG is present, glucose is absent, and X-gal is present, but it will not occur due to the absence of the glucose. IPTG is present, glucose is absent, and X-gal is absent, so the reaction won't occur without glucose, but it also will not turn blue because it doesn't have the X-gal molecule. All of them are present, so it will occur and it will turn blue. And then IBTG, present, glucose is present, but X-gal is absent. The reaction will occur, but it will not turn blue because you are missing the X-gal. So the point of this question is, for the reaction to occur, you need both IPTG and glucose, and for it to turn blue, you need X-gal as well. Okay, and problem 4. So all of them are on, and so it would be a constitutive process because it'd just be continuing. The next one, the I has the defective S. It produces a lac repressor that can bind to the operator but can't bind to the allolactase. So it's inducible because it needs something. It can happen, but it needs help in order to happen. It needs kind of an outside force. The next one's non-inducible because the P, it won't know where to start. And it's got a couple of defective genes. D is inducible. It will occur, but it does, again, need the help because it has that IS gene on the F prime plasmid. And then the last one is non-inducible because it has two defective genes. It has that OC, and so the lac oppressor cannot be removed. Okay.
Awesome. Okay, number 5. Cool question. Once I understood what was going on. So it's talking about will your solution turn yellow or not yellow in the presence of lactose or the absence of lactose according to special genotypes given found within the E. coli here. So it gives us some parameters saying what the symbols mean. And then it asks us to label the search scenarios as 0, 1, or 2. 0 meaning that no yellow color will form. 2 meaning that some will form, and only one copy of the lac operon is transcribed. And then 2 means that it's really yellow, indicating that more than one copy, or in this case, 2 copies of the lac operon were transcribed. So case A is talking about this genotype right here. We notice that immediately the P negative here means that that's defective, and that's for the promoter. So in both cases, for lactose positive in the lactose positive test tube and the lactose negative test tube, transcription will not occur because it won't know where to bind. Okay, in case B, we see that all of the genes are positive for wild type, and that the operator sequence is defective. But it's defective in the case that the lac repressor can't bind to it, meaning that RNA has nothing to get in its way to keep it from transcribing. So maybe because there's only one sequence here, you would only get one copy. But anyways, you see 2 here just for the idea that it will transcribe fully. I think you only get one copy, but in that case, either way. For C, in case C, we see that we have a merozygote form here. In the first copy, in the first chromosome here, we see that all of them are positive, so that will transcribe one sequence there. And then on the F prime plasmid, we see that all of them are wild type except for the operator sequence is defective, meaning that the repressor can't bind, meaning that RNA polymerase can't be inhibited, meaning that you will form two sequences. It will transcribe twice, 2 copies. So in lactose positive, you'll see 2, and in lactose negative, you'll see 2 as well. For the D case, we see that it's another merozygote form, and in the F prime plasmid, we see that P is negative as well. So in this case, due to that, you'll only generate one transcription here, or one product, and the I negative as well means that it's always refers to the defective lac gene. So you'll generate 1 copy in this case, and then in the lactose negative test tube, you won't have any. For the last case, we've got the same setup as in D, but we see that the I gene here is defective. There's something wrong with it, meaning that this IS gene produces the repressor that can bind the operator, but it can't bind to the effector molecule that will help release it off of the operon sequence. So what happens is that you will generate 1 copy from that first chromosome, but you won't get any from the F prime plasmid. So you would get 1. And then in the lactose negative test tube, you won't get any as well. And that is it for our video.